Chapter 13, Viral Replication. The learning objectives for this topic are: sketch and describe the 5 stages of the lytic replication cycle as it typically occurs in bacteriophages. Compare and contrast the lysogenic replication cycle of viruses with the lytic cycle. Explain the differences between bacteriophage replication and animal virus replication. Compare and contrast the replication and synthesis of DNA, negative sense RNA, and positive sense RNA viruses. Compare and contrast the release of viral particles by lysis and budding. Compare and contrast latency in animal viruses with phage lysogeny. In this topic, we're going to cover lytic replication of bacteriophages, lysogeny, and replication of animal viruses. Let's go over this diagram that shows lytic bacterial virus replication in bacteriophages. Now, one thing to keep in mind, especially when we compare bacteriophages to animal viruses, bacteria have a cell wall. Animal cells do not have a cell wall. So that affects how the virus can get into the cell and how it has to leave. So, the first thing that happens is attachment. The phage grabs a hold using its receptors to grab a hold of cellular receptors on the bacterial virus. Now, because of the cell wall, bacteriophages, the capsids, do not enter the cell. They simply can't get in. What they do is they poke a hole in the cell wall and the cell membrane, and they inject their genome. Then they also inject an enzyme that goes and chops up the bacterial genome. Now the cell cannot replicate its own genome. It's all chopped up. But there is some DNA here, and the RNA polymerase gets involved, makes mRNAs. The mRNAs are clamped onto, the ribosomes clamp onto the mRNAs, and you make viral proteins. This is the synthesis phase. Then all of these phage proteins self-assemble, and viral copies of the genome are inserted into the capsid. Now, because of the cell wall, you have to lyse the cell. That's the lytic part of the lytic replication. You have to lyse the cell. It's pretty well dead anyway. If it can't make its own proteins, it can't maintain itself. But we have lysis. You have release of the new phages, which go on to infect a new cell. Before we move on to the next topic, I want to bring up topics from previous chapters just to connect everything. Now, remember that sometimes pieces of bacterial DNA can get inserted into these capsids and go on and inject bacterial DNA into another cell. Because the bacterial DNA is not phage DNA, it can't make the enzymes to chop up the rest of the bacterial genome and other things, and so the cell survives. And we call this transduction. Also, at this point, let's back up over to here. At this point, after the phage DNA has been injected, there's a race that goes on. Who's going to chop up whose DNA first? Is the phage going to chop up the bacterial chromosome, or is the bacteria going to use its restriction enzymes to chop up the phage DNA before it can start undergoing synthesis? So remember, that's where we get restriction enzymes that we use in genetic engineering. Not all bacteria undergo lytic replication. Some undergo what we call lysogenic replication. So let's take a look at a lysogenic viral replication cycle. So once again, we start with attachment. The lysogenic phage, in this case a lambda phage, punches a hole in the cell wall in the cell membrane, and injects its DNA into the cell. Now, it can go on to chopping up the bacterial chromosome, synthesis where it makes its genome and its capsid parts, assembly of everything, and then lysis. But what often happens is, once it injects its genome into the cell, that genome is incorporated into the bacterial chromosome, and we call that a prophage. And it just kind of hangs out. Whenever the cell divides, the prophage is replicated right along with the bacterial chromosome. And so we get more and more cells that have this prophage. So the prophage is being duplicated, is being replicated without killing the host cells. Now, if we have an event that we call induction, the bacterial cell is damaged. The prophage says, "Whoa, this ship is sinking, we better get out." They will remove themselves from the bacterial chromosome, they will chop up the bacterial chromosome and go into the synthesis, assembly, and release so that they can get to another healthy cell. Now, I say that it's just kind of hanging out. An awful lot of lysogenic viruses carry genes that they've picked up from other cells. Sometimes they have a resistance gene. So once they become a prophage in this bacterium, that resistance gene is transcribed and translated, and this bacteria becomes antibiotic resistant. Or it may have a toxin gene that gives this cell an advantage within the community, kills off other bacteria, or turns the bacterium from a non-pathogenic strain to a strain that causes disease in humans. So lysogeny is actually a good thing for the bacterium.